Hello everyone, welcome back to more human physiology, and we are going to now start a chapter that is going to actually cover quite a few different topics. So we kind of throw them all into this category of what we call the somatic nervous system. Well, really the main thing that we're going to be talking about here is afferent signals. So we're going to be talking about all five of our senses in terms of things like touch and taste and smell and vision and hearing. We're going to focus on how sensory neurons and very special sensory organs actually take particular stimuli like the light that we see or the vibrations in the air that we hear, turn those into action potentials, and then how does that afferent information reach the brain and how does it get processed? That's going to be kind of the focus of this chapter. So the next chapter that we'll get to, which is on the autonomic nervous system, and then shortly thereafter in the next week we will talk about muscle physiology, we'll be focusing a lot more on the efferent side of things. So once we have decided what our course of action is in our control center, how do we get that integrated information delivered to our effectors? So the topic du jour here is going to be sensory neurons and afferent messages. So I'm showing this particular slide again. We've actually looked at this before, just as a reminder that generally speaking, any type of neural pathway that utilizes the spinal cord is going to look almost essentially like this. So this was the example of someone tickling the bottom of your foot with a feather. Well, back in chapter 13, we covered how does that afferent information get sent into the dorsal root of the spinal cord, up through ascending tracts to the medulla. It crosses over to the contralateral side at that point. Then it goes to the thalamus, and then it goes to the appropriate part of the cerebral cortex, which is going to be the somatosensory cortex and the parietal lobe. So that's all stuff that we've covered before, and all that stuff is still going to apply here for certain afferent messages. So we're not necessarily going to cover the efferent side of things here quite as much, but that information will be coming later on. Okay, so let's talk about the somatic nervous system. So in the previous example, we talked about the case of someone tickling the bottom of your foot, or if maybe it kind of fits into your worldview a little bit better, you could maybe talk about touching a hot stove, right? So you touch a hot stove like what you see here. It's very hot. Obviously, your natural inclination, totally involuntary, is going to be that you jerk your hand away. That's why I have voluntary in quotation marks here. It's not exactly voluntary, right? So on the previous slide, basically the exact same sort of thing would apply. You're going to stimulate sensory receptors in your skin of your hand that you're using to touch the hot stove. That afferent information is going to go through your arm, into the dorsal root of the spinal cord, up ascending tracts to the brain, and then your brain is going to basically come up with the bright idea of, hey, maybe we shouldn't have our hand on the hot stove, just spitballing here, right? So the brain decides we need to get the hand away, so it's going to send the efferent message back down through the descending tracts of the spinal cord, out through the ventral root, and to the muscles in your hand and your arm so that you can jerk your hand away. So that is an application of what we would call the somatic nervous system. Essentially all of the both voluntary and involuntary things that you are at least consciously aware of. Now, our focus on the autonomic nervous system in the next chapter is going to cover involuntary activities that you are not yourself aware of. So that discussion will be forthcoming. So in the case of touching the hot stove, you are activating a particular type of sensory receptor called thermoreceptors, and we'll talk about all the different types later on. So activation of these receptors leads to a very strong graded potential. We call that an EPSP, if you will recall. That should get us to threshold, so that will give us an action potential that is carried through the axon of the sensory neuron into the dorsal root of the spinal cord. And if we need it to, we can send that message up through ascending tracts to the brain. But of course, in the previous chapter, we looked at an example of the patellar tendon reflex in which it's very quick, it doesn't go to the brain, it just goes in through one side of the spinal cord and out through the other. Speaking of going out through the other, the efferent signal will be sent immediately as an action potential through a motor neuron, and its axon projects out through the ventral root of the spinal cord. And in the case of the somatic nervous system, your effector is almost always going to be skeletal muscle, almost always. So any other type of target like cardiac muscle or smooth muscle, sweat glands, endocrine glands, etc., that is almost always going to refer to the autonomic nervous system, not the somatic nervous system. So when you see the word somatic, think skeletal muscle. That's kind of the end game there. So as I've said, our focus here is mostly going to be on the afferent side of things. So everything from the time that the stimulus is presented to us, we sense it, to the time we deliver it to the central nervous system. That is the definition of afferent as we understand it. So when a particular type of sensory receptor is activated by a particular stimulus, this is going to lead to a graded potential. In the case of sensory neurons, we call these graded potentials either generator potentials or receptor potentials. Now, don't let those two new terms fool you. The graded potential is still exactly the same as we understood it from Chapter 12. The stimulus still needs to reach a certain threshold to give us an action potential as we discuss. So nothing really has changed much there. So assuming that we do get an action potential, it will be conducted in the direction of the CNS, but the route that we take to the CNS, whether it be through a spinal nerve or through a cranial nerve, is very much going to depend on what the stimulus was. And that will lead into our next discussion in the next video on general versus special senses. Now, something that is very necessary for sensory receptors is that we have to have a signal transduction mechanism. So we talked about signal transduction in our communication unit, right? So the idea here is that whatever our stimulus is, that stimulus is likely not electrical in nature. The action potential is electrical in nature. So what we need is some way to transduce or change the stimulus so that it does become electrical in nature. Basically what we mean is whatever the stimulus is, it needs to lead to the opening of ion channels on the sensory neuron. We have to find a way to turn our stimulus into a graded potential. And our stimuli are going to come in many different forms. It may be light, it may be changes in pressure, it may be changes in temperature, it may be the chemicals in the food that we eat or the things that we smell, the vibrations in the air that lets us hear things. So our stimuli are going to come in many forms, and we are going to have very special mechanisms for turning those stimuli into graded potentials. So here you can see several different types of sensory neurons. The idea here is that most sensory neurons that we understand about, the most basic type, contain their sensory receptors in the dendrites, and sometimes these dendrites are called nerve endings. So these nerve endings may or may not be encapsulated by a protective layer of connective tissue, so you can kind of see both cases there. But in either case, you'll notice that these sensory neurons are, as we typically understand them, to be pseudo-unipolar, meaning that these cell bodies, as you see here, provided that they project into the spinal cord, those cell bodies are going to be found in the dorsal root ganglion. Now, look at this sensory neuron down here. That's a funky-looking sensory neuron. Actually, it may not even be accurate to call it a sensory neuron. What may be more accurate is to call it a specialized type of sensory cell. So what you're looking at here is a light-sensing cell called a rod photoreceptor. Basically, when our stimuli become especially unusual or atypical, like light or like vibrations in the air, we need these specialized sensory cells to be able to take that unusual stimulus and produce a graded potential. So this photoreceptor is a good example of that, and then that sensory cell will typically synapse on a cell that actually delivers the afferent message to the central nervous system. In this case, you can see a bipolar cell here, which will connect to other cells in the retina, and we will talk about that towards the end of the chapter. So, like I said, these specialized sensory cells are typically going to be found in very specialized sensory organs, like the retinas, the inner ear, the cochlea of the ear, the taste buds, the olfactory bulbs, etc. So you're typically only going to see your more typical sensory neurons when you're talking about your more general senses like touch and temperature and things of that nature. Okay, so while we're still on the topic of sensory receptors, let's talk about two different types of receptors. We're not talking about types of sensory neurons, we're talking about types of receptors regarding the stimuli that we respond to. So, phasic receptors and tonic receptors. A phasic receptor is going to be one that adapts to the stimulus while the stimulus is still applied. Well, that sounds like a whole bunch of scientific mumbo-jumbo. What's kind of the 411 there? Well, basically what a phasic receptor refers to is something that you can get used to the longer that you're basically confronted with it. Perfect examples are temperature, odor, and touch. So, especially temperature and odor. So, one of the first labs that you did this semester was had you stick your fingers in warm water for a few minutes, and then you kind of felt like you got used to it, right? And that's because the set point of your skin temperature receptors rose to meet the temperature of the warm water. That's why you feel like you get used to warm water when you take a bath or you take a shower, right? So the idea behind something like this is that from the time the stimulus is applied, assuming it's a fairly strong stimulus, the strength of that stimulus, as we've discussed, is indicated by the frequency of action potentials that are fired through that particular sensory neuron axon. But if the stimulus is applied for a good length of time, eventually the receptor's set point is going to change depending on what the stimulus is, and the closer the set point gets to what the stimulus actually is, the smaller and smaller the graded potential is going to be, and the fewer and fewer action potentials are going to be fired. So this is why you feel like you get used to a certain temperature, or you don't notice the rank smell of fast food in your car anymore. The longer time you spend in the vicinity of the stimulus, the less and less you're going to feel like there is a stimulus at all. And this is because that sensory neuron axon fires fewer and fewer and fewer action potentials over time. Now, the other example is what's called a tonic receptor, and really tonic receptors only refer to one type of sensory modality, and it's about the worst one you can imagine: pain. No matter how long you have been injured or something has been hurting you, pain is pain. You don't ever get used to pain, and that's because pain receptors, or what are called nociceptors, are of the tonic variety, meaning that there is no adaptation. The only way pain can be alleviated is absent of taking painkillers. The only way a pain stimulus can truly be taken away is if the injury heals or whatever is causing the painful stimulus is removed. As long as that painful stimulus is still there, as long as it is there, the action potentials will be fired at a very constant rate. And like I said, the only thing that can really take that away is to actually fix whatever is causing the problem, meaning the injury. Another way that we can classify sensory receptors is based on the location of the stimulus, and this is going to be the classification method that is going to be the most useful for us, because basically the way that we do this, it's going to tell us exactly what the stimulus is. So, generally speaking, we can refer to these as either exteroreceptors or interoceptors. Basically, it's going to tell you where's the stimulus. So, as you can kind of tell, you can kind of see the root of external and internal in these words. Exteroceptors are going to be receptors that are positioned near an external stimulus, meaning that temperature or touch receptors on the skin are going to be activated by outside forces, such as someone slapping you on the wrist or burning your hand on the stove or something like that. Interoceptors are receptors that are positioned near internal stimuli, meaning these receptors are kind of pointed at the internal environment of your body, and these are going to be a lot more useful for whether it be special senses like vision or hearing or things of that nature, or for more autonomic things like regulating your blood pressure and things of that nature. So, like I said, a perfect example of this are the baroreceptors or pressure receptors that detect changes in your blood pressure and allow your autonomic nervous system to make adjustments on the fly. Proprioceptors are going to be receptors that are near moving body parts. They're going to be kind of in the neighborhood of joints and places like that. So, perfect example of this was the Golgi tendon organ in the patellar tendon reflex. It measured the amount of stretch being placed on the patellar tendon, basically allows you to know which muscle groups to be activating depending on the amount of stretch in the knee that you feel. Okay, so on the previous slide when I said this is going to be the most useful way of classifying receptors, this slide is what I meant it for, so I apologize for the confusion. Classifying sensory receptors based on what the stimulus is by far going to be the most useful thing that we can do here. So basically the question we're asking here is ultimately what causes the sensory neuron action potential to be fired, right? So these are the general categories here. We have chemoreceptors, which are going to involve receptors being activated by a chemical stimulus, meaning some type of molecule like food or odorant molecules, gases like oxygen and carbon dioxide. Photoreceptors, as the name implies, are activated by light stimuli. Thermoreceptors, again, as the name implies, are going to be activated by changes in temperature. Mechanoreceptors will be activated by touch and pressure and vibration. Osmoreceptors, like those you find in the hypothalamus, are activated by changes in solute concentration. So we've already seen how osmoreceptors are a part of the neuroendocrine reflex involving the release of antidiuretic hormone from the posterior pituitary. And we'll certainly talk more about that when we get to the urinary system. And then I already mentioned nociceptors, which are going to be activated by pain. Well, pain is kind of a weird, kind of an intangible thing. So the specific thing that these are activated by is tissue damage. So when you hurt yourself, it's because you have damaged your cells and damaged your tissues, and those cells release factors which activate these nociceptors. But the funny thing is, not all receptors are necessarily created equal. So here's what we mean by that. So we just classified these receptors based on what the stimulus is. So photoreceptors are activated by light, chemoreceptors are activated by chemical stimuli, etc., right? So general idea is each type of receptor is going to have its kind of preferred stimulus, right? But that does not necessarily mean it is the only thing it can be depolarized by. So when we talk about the preferred stimulus, we're referring to what is called the adequate stimulus, meaning the most readily accepted stimulus for each type of receptor, and it's just what we said. Photoreceptors, their adequate stimulus will be light. This table here calls it the normal stimulus, but it's the same thing. Chemoreceptors, it'll be dissolved chemicals. Pain receptors, it'll be the chemical factors that are released by cells when they're damaged. So those are the adequate or normal stimuli, but we can activate those receptors with another type of stimulus provided that that stimulus is strong enough. And a good example of this is when you inadvertently, of course, take a blow to the face, a blow to the eyes. Like say maybe you kind of turn your head really fast and you run into something, right? So you kind of wince, you close your eyes, you kind of hold your face, and you kind of see stars, right? You kind of see these kind of bright lights flashing in front of you, even though your eyes are closed. Well, the question is, what's causing the appearance of those lights that you're seeing, the stars that you're seeing? Your eyes are not open, so certainly they're not caused by the activation of photoreceptors by light. Well, what happened here is that the photoreceptors took such a strong mechanical force, meaning the blunt force trauma to the head, that they actually activated and started firing afferent signals through the optic nerve. So that's what causes the seeing stars phenomenon that I'm sure you're familiar with. All right, so that's going to do it for this little introductory video into the somatic nervous system. Join us next time and we will have a very short discussion on the difference between general senses and special senses, particularly how these types of afferent messages tend to be sent into the brain. So I hope you'll join us next time.